Okay, so good afternoon. It's my pleasure to be here today, not only in the context of Human Glycome project, which was really interesting, but also in the context of our GlycanSwitch project. So I'm Inês Alves, and I'm a postdoc at I3S, and I'm just going to make a pause here in the IgG glycosylation and show you a little bit of the work that we have been doing in the past years, and then introduce you a little bit more again in the IgG and autoantibodies. So our prototypical model and a very attractive model for studying autoimmune diseases is lupus, because it's a systemic autoimmune diseases, and I don't need to introduce a lot because Klaus did very well. But one of the most severe complications of lupus is kidney disease and lupus nephritis. Up to 50% of the patients can actually develop lupus nephritis, and from these patients, up to 25% in Europe at least can progress to chronic kidney disease, leading to the need of hemodialysis or even renal transplantation. So up to now, there are no very nice therapeutic approaches. Actually, the goal for clinicians is treat to target, which means achieve remission or even low disease activity, but the drugs are available are still associated with comorbidities and even very low rates of response by the patients. So we need novel target therapies, but we need even more to understand why this loss of tolerance occurs. There are a lot of evidence already on the mechanism behind the loss of self-tolerance in autoimmune patients, and we are very interested in studying how altered self-antigens can actually occur at the post-translational level. So, as we all know, protein glycosylation is a major post-translational modification that occurs in all living cells, and we are studying and focusing on the N-glycans that occurs on the asparaginins and are the major glycans presented at the membrane proteins. So actually these glycans are produced and synthesized between the ER and the Golgi by the consecutive action of glycosyltransferases and glycosidases, and it gives a rise to a very diverse and complex repertoire of glycans. And I don't need to convince you that glycans are important because Gordon does it very well, but I just found this picture very interesting. So we can see here the spike protein of SARS-CoV-2 virus, but actually it occurs naturally with all these glycans in dark blue. So we almost do not see the protein skeleton. What we see is the glycans. And also, at the cellular level, the glycocalyx is the first layer between the interface of two cells. So as you can imagine, glycans are very important for this cell-cell interaction. And glycans are also associated with the evolutionary process. So less complex glycans are usually found in microorganisms, highly manosylated, pauci-manosylated, and we mammals are more elongated and more complex glycans. So we avoid this accumulation of mannose residues, and we are able to synthesize and elongate our glycan conjugates. So just as an example, how glycans are important for their recognition by the immune system. For example, HIV virus that is covered with these high mannose and glycans, the MAN9, for example, is able to be recognized by DC sign, a C-type lectin that is expressed by innate immune cells and triggering a Th1, Th17 pro-inflammatory response in the immune cells. So these glycans are also important to be sensed by the immune cells and trigger these pro-inflammatory responses. So we hypothesized that lupus nephritis and this loss of self-tolerance at the kidney parenchyma could be associated with an altered glycosignature at the epithelial compartment. So in collaboration with three national hospitals, so Santo António, Coimbra, and São João in Porto, we were able to perform this 3D mapping of the N-glycosylation profile by mass spectrometry imaging with the collaboration of Manfred and Bram from LUMC. So we also validated these findings by lectin binding in situ and also flow cytometry analysis. And after 1 year of follow-up from these patients, we were able to collect clinical variables and correlate with our findings. So as you can see here in this map in blue, you can see the glycosignature of the samples from healthy kidney, and in pink we can find the lupus nephritis kidney signature. So it seems different. Actually, it's different because there is an increase on these mannose-enriched glycan traits and a decrease on the complex N-glycans. And we can specifically assign that these main differences were contributed by the epithelial compartment, so the glomeruli and the tubuli of the kidney. And one of the most pinpointed or one of the most top glycans that appear to be increased is the MAN9. So actually a very similar microbial related glycan. We validated these findings in lectin histochemistry by LPHA and GNA lectin. And we found the same trends. So there is a decrease on, the complex glycans on lupus nephritis tissue and an increase on the more high mannose glycans in this disease kidney. We also observed that there is this increase on the DC sign expression binding motifs from the kidney. So it potentially suggests that DC sign can be recognizing more this tissue than, for example, the healthy kidney tissue. We saw that these differences were specifically for lupus nephritis kidney and not other kidney diseases, as it may be associated with just disease state. So, interestingly, for the clinical purpose, we were also able to test if this marker, so high levels of mannose, was associated with bad prognosis. So bad prognosis for these patients is mostly the development of chronic kidney disease at least 1 year of follow-up. And we observed that high levels of high mannose were associated with the development of chronic kidney disease statistically significant. So we found this, we characterized this glycosignature from the kidney from the patients, but we also wanted to understand if this signature, this glycan, could be potentially triggering an immune response contributing to the pathogenesis of lupus disease. So we saw that associated with this glycan signature that you can see here, this is kidney from lupus nephritis patients and this is kidney from other kidney diseases, we saw an accumulation of a specific subset of T cells. So it's gamma delta T cells that usually are in the bridge between innate and adaptive immune response, so they are not dependent on the MHC presentation, and also they show and they express very related receptors to the innate immune cells. So for this, they were actually very interesting to find, and we did not observe differences on the alpha-beta conventional T cells in the kidney. So gamma-delta T cell infiltration is associated with levels of mannose on this tissue, so it seems to be association between glycosylation and the presence of these cells. And moreover, we also found an increase on the DC sign receptor. So as I told you, DC sign is able to recognize these high mannose and glycans and trigger an immune response. So it was increased on the lupus nephritis samples compared with other kidney disease. We took advantage of our mice model that is knockout for MGAT5. And MGAT5 is the gene that codes for N-acetylglucosamine transferase 5, so glycosyl transferase 5, and it puts like the last branch of the tetraantennary glycan on the glycan. So we saw that with age, these mice accumulate high levels of high mannose in the kidney, and this actually seems to be associated with the infiltration of immune cells. So there is a glomerulonephritis that happens in these mice spontaneously, that has been already described by Dimitriu. And we saw that these high levels of hymenos were also associated with these SINR3 binding motifs, which is an homologous of the DC sign. Spontaneously, these mice appear to have like lupus-like syndrome with proteinuria, high levels of autoantibodies, and even nephritis score. And by characterizing the immune cells of the kidney, we saw an infiltration of gamma delta T cells, more specifically the ones that actually produce IL-17. So as a proof of concept, we just crossed these mice with the knockout for gamma delta T cells to check if we could inhibit this immune response. And we saw a decrease on the nephritis score together with a decrease on the proteinuria and also the production of autoantibodies and pro-inflammatory cytokines. So I've just shown you that kidney from lupus patients seems to be changed, and this can be another mechanism that can also explain the loss of self-tolerance or contribute to the loss of self-tolerance. And now we are moving on to try to understand if we can find an anti-glycan antibody that can also help to monitor these patients in the circulation, and that can be specific for lupus nephritis. So anti-post-translational modification antibodies are not new. They have been described for many years ago, and as Salome presented yesterday, we can actually find anti-Saccharomyces cerevisiae up to 6 years before the establishment of Crohn's disease. And these are not bystanders antibodies. They are actually pathogenic, and they can actually influence in the development of the disease. So in lupus, we tried also to check if we could identify antibody that could react against glycans. And in collaboration with rheumatology, René and Yolene, and also the nephrology department from LUMC, we were able to test in a very specific two cohorts, Symbiose and Aurora, which are cohorts that comprise samples from lupus nephritis patients naive to the treatment. And we were able to see that, in fact, lupus nephritis patients develop anti-glycan antibodies, increased levels of anti-glycan antibodies. Interestingly, the levels of the anti-glycan antibodies seem to be associated with the expression of this glycoantigen in tissue from the same patient. So it seems to be an association between the anti-glycan autoantibody and the presence of the glycoantigen. In a more unbiased and exploratory approach, we came to SIC Cartuja and SIC Biogun in Spain to these chemistry labs, and we were able to synthesize a glycan library, a diverse glycan library with different glycans with different valent presentations, and we printed these glycans on a glycan array with the collaboration of Padler Caravani lab in Tel Aviv. So we saw that in lupus nephritis, and specifically in the patients that have actually nephritis and not other lupus, we saw an increase on this anti-glycan antibody that was statistically significant. And we also saw the same trend for the preclinical samples from Crohn's disease. And more important is that we can actually see a trend to increase along the establishment of Crohn's disease. So moving now to more specifically Akpa, which is also post-translational modification, and the GlycanSwitch and the RA. René and Tom and Johan, they show that these Fab glycans, these Fab from Akpa are actually rich on Fab glycans. So this Fab glycan seems to be associated with the pathogenesis of RA, and we are trying to understand how these Fab glycans contribute to the selection of pathogenics and autoreactive B cells during this process where B and T cell actually interact. So Rafaela Nogueira, which is a PhD student from GlycanSwitch, she has moved to SIC Biogun in Bilbao, Spain, to try to understand which are the glycan binding proteins that can be mediating this interaction or influencing or inhibiting also the interaction between B and T cells along with the Fab glycans. So she has been collecting a library of recombinant glycan binding proteins such as, for example, galectin and Siglex, and she is going to attach the B cells to the sensor and then test which are the GBPs that are found to have differences between the Fab glycosylated and the Fab non-glycosylated B cells. And after this, we just identify these top GBP candidates and move on to the functional in vitro co-cultures of B with T cells. So finally, we are also generating this mice model together with René and Tino from Orpit, right? And we have good news. Actually, the mice have been successfully transfected, as we can see here by the human light chain. So these mice, I believe that they will arrive until the end of the year, and we will be able to characterize if de novo glycan sites are arised, but also how these cells can get an advantage to this selective process during the BT interaction. So finally, I just want to thank to everybody that has contributed to the work, the work that we have been developing through the years, but also that have been involved in this last year's GlycanSwitch work, and of course to the funding agencies and all the patients that contributed. Thank you.
Thank you for the on-time questions of the audience.
English, this was a surprise that you went down with the mannans or the glycans, let's say, which are targeted. So what are now the specific motifs which you find, which are bound by the antibodies?
Yes, so this was a very high throughput analysis, so we are still analyzing. We just combine all the glycans of interest, which I think you may know which are. So we just put them all together, complex glycans, because we have mono presentation, but also multivalent presentation, and it's also changing. And we just put them all together, and we put the other ones as non.
Okay, so you have a subset, but do you have a defined motif?
Yes.
Sure. Okay.
Yeah, we have done it like glycan by glycan, so we know exactly which are the glycans that are contributing to this subgroup.
Thank you, Inês. I think I have a similar question. How the anti-sugar antibodies, glycan antibodies, do you have any idea how they are induced?
Yeah.
Recognized it.
Yeah, so they have been, for some years, they have been studied in the context of extrafollicular reactions and more IgM, specific IgM. But we also have natural IgG antiglycan antibodies. And Akpa, for example, is one example that it's an antiglycan antibody. Of course, that it's from a microorganism, or maybe not, we don't know. But probably along with the time, because we also see here, so healthy controls are not negative.
So we all have them.
We all have them. I think it's just a matter of abundance. And I also see it in the glycan array, which was completely different samples and completely different setup. I also see that healthy controls also have them. And we probably have them from the beginning that we are born, and then it's just a matter of antigen presentation and production of these antibodies.
Other questions? If not, thank you very much.